Hi, my name is Quinn Montgomery. I'm a PhD student in Andrew Barton's lab, and today I'll be talking about environmental change shapes phytoplankton morphological traits in the coastal ocean. This work has been a collaborative effort with myself, SOSIC Lab, and Andrew Barton, my advisor. The first thing I want to talk about is when we look at these IFCB images, this is going to talk about IFCB work, keeping with the theme. But when you first see these IFCB images, we can see that the size of phytoplankton is the number one thing that really stands out to us when we look at this the traits of these images of these phytoplankton cells on the right. The size of the phytoplankton are hugely important to defining their physiological, biological interactions and their fitness. This image shows the phytoplankton cells sampled by an image flow Cytobot, and the most apparent trait variation is the really high degree of variability of cell size. Phytoplankton size structure is a key characteristic in marine ecosystems. That changes in the structure can have large impacts on trophic characteristics like trophic structure. Marine community with larger phytoplankton cells will have fewer trophic transfers and will more efficiently transfer energy to higher levels in the food web. And then it will also have effects on sinking rates and carbon export. Larger cells will more readily sink out of the surface ocean, sinking at much faster rates than smaller cells. Large diatom can sink on the orders of meters a day, where small prochlorococcus cells will be in order of millimeters. So it's important to study the kind of size characteristics of ecosystems because this can have effects on how they both the environment and sequestration of carbon. So here I show a metric we use for estimating the size distribution of cells in the phytoplankton environment. Generally, in aquatic environments, size and abundance are inversely related, meaning that larger organisms exist at much lower abundance than smaller organisms. And this decrease in the size abundance relationship can be drawn as a log linear relationship, as shown on this plot on the right. So this is showing a full marine ecosystem with green being the background phytoplankton, blue being the zooplankton, and red being the fish community. And so we can define this specific relationship. I'm particularly interested in phytoplankton, but this relationship can also be applied to many different organisms within the size spectrum. And it's specifically defined as the abundance size spectrum, and is calculated as the log-log relationship between the abundance of cells within a defined size interval and the size of the defined interval. And so it's important to look at because this relationship can be drawn linearly from the very base of the food web and through higher trophic levels. So impacts in the very base within the phytoplankton can have ramifications for higher trophic levels. And the changes in the slope can have implications for the storage of carbon in the ecosystem, and as I talked about, higher trophic transfers. So if we were to think about this slope being steeper, there would be a greater abundance of overall smaller phytoplankton, greater overrepresentation of smaller phytoplankton than larger phytoplankton. So here I show two plots showing the regional variability in phytoplankton abundance size spectrum. The top plot is the phytoplankton size spectrum in the ligotrophic Atlantic very ligotrophic areas of the subtropical gyres in the Atlantic, and the bottom plot is taking the phytoplankton size spectrum from an upwelling zone along the Iberian Peninsula. So very generally, when we measure these, we can see that the slope of these phytoplankton size spectrum differs between these two regions. This is from Marinon et al. 2010. So the oligotrophic region has this very steep slope because there's fewer nutrients available for these phytoplankton, and the larger phytoplankton are outcompeted. But when we see in the bottom plot here, in the coastal upwelling zone, we see a greater proportion of larger phytoplankton represented, which is due to the increase in nutrient availability. And so obviously there's some suggestion that this phytoplankton size abundance spectrum varies spatially. And so here I show another plot of regional variability in phytoplankton size spectra from a global size structured phytoplankton model. A more negative slope in blue indicates a phytoplankton community that is more dominated by small organisms. And so we can see that there is this real great spatial variability from this model output, showing that there's not very steep slopes, less steep slopes of the phytoplankton in the very high latitudes, and in the subtropical gyres, we see this very very steep slope occurring because there's generally less nutrients available. And the findings of this model generally predicted that the phytoplankton size spectrum changes in result from environmental drivers, including nutrient availability and grazing control. So my general question is, so we've seen evidence that the phytoplankton size spectrum changes over spatial variability, but my question is, does the phytoplankton size spectrum change over time? So we've seen that community composition is clearly linked to seasonal environmental drivers, and we would expect to see seasonal changes in the distribution of biovolume between size classes in a very temperate environment. So to measure this in keeping with the previous talks, I used this tool called the Imaging Flow Cytobot, and I'll move through this a little bit quicker because obviously there's been a lot of talk already about what this instrument is. But it's this very, very cool instrument. It's essentially just, well, as we talked about, it samples images of cells, and it can measure cells between about 10 microns and 150 microns in cell length. This picture on the right is an IFCB that is currently deployed off the coast of California in Del Mar, and that's a picture of the inside of the instruments. And to get a little bit into this, we've talked a lot about this already, so I'll move through this quickly. This is the general image output we get from the IFCB. And you can see just from this image output itself, as I mentioned previously, that there's this really nicely captured cell size structure in this environment that the instrument does a really good job of taking and quantifying. And so the study site for this analysis was Martha's Vineyard. So Martha's Vineyard is a very highly temperate environment, and this was deployed since 2006. So this data set, as I used the data set from 2006 to 2018 for a total of over 260,000 injections or samples into the instrument, or more than 800 million cells sampled in total. So quite a powerful data set, and it was really useful for this analysis. So Martha's Vineyard Coastal Observatory, or MVCO, is a very temperate coastal location with high seasonality. Here I show a seasonal climatology of the various parameters of interest I used in this analysis. These plots show the average value of each month of the year, with the shaded areas indicating standard deviation. Generally, in the winter months, the water temperature is much cooler and the salinity is higher and wind speed is greater, all factors which I imagine would help increase the amount of nutrients in the study area. On the bottom plot, I show seawater sample data of chlorophyll A and nitrate concentrations. Generally, we can see that with these, this is just bottle sample data, with increased nutrient concentrations, we see this kind of general shift in the amount of biomass in the area as recorded by chlorophyll A. And also, the position of Martha's Vineyard Coastal Observatory makes it sensitive to oscillations within the NAO, or the North Atlantic Oscillation, which is the dominant mode of atmospheric fluctuation over Eastern North America and the North Atlantic Ocean. Positive NAO at MVCO is generally associated with warmer winter air temperatures and increased precipitation. And then a negative NAO is generally associated with colder winter air temperatures and stormier conditions. So there's this, I used the Hurel monthly PC-based NAO index in this analysis, and you can see generally over the course of the time series, the NAO has been mostly positive with periods of it becoming negative. So to get into my analyses a little bit, here is a kind of a top-down look at the phytoplankton size structure at Martha's Vineyard. The color indicates the percentage of cells in a given day falling within each volume bin, and the sum of each column or day is 100%. So basically, color is just indicative of where most of the biomass is. And then the thick black line at the bottom shows the daily median biovolume. And in this analysis, the cells are grouped into size bins of 100 cubic microns. So as you can see, there's a very high degree of seasonal variability within the phytoplankton size distribution. Very large phytoplankton only make up a measurable percentage of total biovolume during the winter and early spring. And in the summer months, we generally see very few or not at all we very rarely see these like very large phytoplankton cells. And you can see that also represented in the daily median cell size, which tends to be much smaller in the summer and peaks in the winter and early spring. So this is a kind of a useful way to look at size structure over the course of the time series. However, I chose to calculate the phytoplankton size spectrum because it allows kind of a more holistic analysis as opposed to just looking at the size distributions. I can calculate the size spectrum as a means of just a single value to determine that what the size structure of the community generally is. So to calculate the phytoplankton size spectrum, I included this little slide because there's multiple ways to do this if you've ever dived into the literature. For my analysis, I binned cells into log space intervals of 0.025 cubic microns. And because I binned into log space, and when transformed back into normal space, that'll result in varying bin widths. I also normalized each bin by the non-log transformed interval width. And so in general, the slopes of this analysis will be around -1 to -2.5, and that's varying from the previous slopes I reported because of this normalization process I used. And then you can see on that plot on the right, I show the slope for a single day at Martha's Vineyard for all the cells in a single day. And I then fit a line to it and calculated the slope for that specific day. So in response to the seasonal drivers I discussed, as I showed, I estimated the daily slope of the phytoplankton size structure, and here I plot the slopes as a time series on the right. As you can see, there is a great degree of seasonal variability over the course of the time series, and there's clear shifts in seasonal size structure. So the slopes, if you look at this analysis, the slopes generally tended to be less steep during the winter months and steeper during the summer months or late spring to summer. And so I was curious about this trend, and to confirm it, I constructed a cyclical generalized additive model of the seasonal variability within the spectral slope. So this plot on the right shows that GAM climatology. Basically, from January through early spring, we see that the slopes are much less steep, and then coming into the summer months, we see that these slopes become much more negative, and then back into the late fall and early winter, the slopes gradually become less negative again, and that will have impacts on how the phytoplankton community engages within its environment. It allows these winter communities will allow for more efficient transfer of energy to higher trophic levels, with these food webs being shorter, and a greater proportion of the carbon will be stored within the larger cell sizes. So it'll have impacts on how this phytoplankton community transports and stores carbon. So very clearly, the size structure changes seasonally, but I was also curious as to what was specifically driving the size structure. And here I've shown the seasonal variability in the spectral slope, but when I was curious to see what was driving this, the variability, I used a technique in decision tree regression called random forest regression to rank the relative importance of environmental drivers. We can rank relative importance of each predictor variable by determining the percent increase in mean squared error, or MSE, of the model when the predictor variable is randomly permuted. So a greater increase in mean squared error indicates a greater predictive importance of the permuted variable. In this case, when I permuted each variable sequentially, I then found that temperature when the temperature was randomly permuted, it was had the greatest increase in mean squared error for the data set, making it having the greatest overall predictive importance for this model. So that's most likely due to water temperature being linked to nutrient availability. Generally, when the water was colder, there was most likely less stratification and a greater intrusion of nutrients into the study area. The second most important indicator, which I found was interesting, was the NAO index, which might indicate some sensitivity of the phytoplankton size structure to NAO, which I found was very interesting. And then the third most important was the salinity, which could be both an indicator of if advection of new phytoplankton communities into the study area, or also some stratification if we see large rain events. And then, interestingly, total biovolume, which I calculated as cell biovolume concentration as measured by IFCB, and solar radiation had only modest ability to predict the size spectrum slope. So this is interesting in that while we do see the seasonal shift in total biovolume and obviously solar radiation as well, this was predicted to have very little or only a modest ability to impact the size spectrum. And lastly, wind speed was overall a poor predictor. I had assumed that maybe wind speed might have something to do with breaking up the stratification and mixing, but it was only a poor predictor of size spectra. So in that, it was interesting. And so here I asked, like, well, how do each of these predictor variables impact the size spectra itself? Here I show in this plot the marginal effect of temperature within the random forest model. Essentially, I artificially manipulated the predictor variable, in this case temperature, along the range of observed values and find the average predicted value for the size spectra slope at each temperature value. This is useful as it shows how the model is predicting the response of the phytoplankton spectra slope to changes in temperature independently of other predictor variables. So generally with this plot, it is showing that as seawater temperature above about 7 degrees Celsius, as seawater temperature increases, the spectra slope decreases. So this is it's useful because we can see that independently of other predictor variables used in the model, we expect to see steeper slopes in warmer seasonal environments at Martha's Vineyard. And in conclusions, the phytoplankton abundance size spectrum varies on strong seasonal cycles, which is very interesting. We can see this strong seasonal change, and happens year after year. And this might have implications for modeling phytoplankton size structure, or just generally for observing these events. And then changes in phytoplankton size spectra within temperature and with temperature and NAO might indicate sensitivity of the phytoplankton community structure to warming from climate change. So we've shown that there's this obvious, we see these effects within the random forest model, which is interesting because I had not necessarily predicted that. Thank you very much. I'd like to acknowledge Heidi Sosik and her team, who both developed the IFCB and oversaw its deployments at Martha's Vineyard. It's really an awesome instrument, both in the original term. It's produced this huge data set, and it's really interesting to see that they were able to basically put what amounts to a microscope and a flow cytometer and have it in the ocean for 14 years at a time. So it's really a cool data set to work with. And then I'd also like to thank my advisor, Andrew Barton, and my lab mates for their help with this project. And with that, I'll take questions.
I'll echo just how amazing IFCB is. It's great. Nice to see all the work that can get done with it. So questions for Quinn?
Fascinating talk. I was really impressed at the work that you did here and relating this change in the spectral slope with environmental variables. I was curious about the total biovolume though, because we typically think of the total biovolume shifting more of the intercept rather than the slope. I was wondering, what you thought about that, and also whether you'd looked at the influence of different taxa, because we saw from previous presentations that there's a pretty good predictor or a automated system for getting taxonomic identifications. So I was wondering if you looked at the taxonomic contributions to the slopes.
Well, generally we see a high degree of diatoms in the winter months. It's a very seasonal environment, so definitely diatoms and these very larger dinoflagellates contributed to it. I didn't actually do an analysis among looking at with the classification of these and relating it directly to the slope, but I imagine it would be highly associated with diatoms and dinoflagellates. And so we do see seasonal variability in the intercept and within nonlinearities in the size spectrum, which I didn't present about here. But I would imagine that obviously the size abundance relationship or size spectrum is going to be very associated with the biovolume, and especially when you include intercept. But I did not relate that in my random forest model.
So we have an online question. So Ken, please unmute and.
Yeah. Hi, I'm Ken. That's an amazing data set you have there. I was wondering, well, you thought about analyzing with other things than just a linear fit to the slope. The reason why I'm asking is, for example, in your example, it looked like there was some kind of scaling, but then the size spectrum was ending. And also on the slide with all the colors where you can see the whole size distribution, it seems there was a big variation actually in the maximum size. So my question is whether you consider others and whether a lot of what you're seeing is maybe not so much changes in the slope, but rather changes in the maximum size.
That might be the case. I did look at I didn't change the way I evaluated the size spectrum. I always used the linear model, but I did evaluate non-linearities within the size spectrum, and that tends to happen on a seasonal cycle as well. So we will see that in the winter months, I showed my size spectrum. I didn't want to pick the best one possible. I had kind of I was like, okay, I'm just going to choose the size spectrum, whatever one is on January 1st, 2010, and that ended up being a rather non-linear day. But so generally we see these non-linearities occur more in the winter months, and in the summer with these smaller, steeper size, with the steeper slopes, we generally see increased linearity.
Yeah, I would think an analysis where one has both the slope and a cutoff would be interesting, because exactly the appearance or non-appearance of large ones are very important, as you mentioned. Thanks.
Thank you.
Other questions?
You may have said this and I missed it. It looked like the slopes that you have at Martha's Vineyard are considerably steeper, generally speaking, throughout the year than either of the data sets. Was it Marignan that you showed earlier?
Yeah, I showed Marignan and Andrew's model from 2013. But yeah, so it's generally sensitive to the way you calculate the slopes. So I use the normalized abundant size spectrum, which basically the slope is kind of almost a function of your binning process and also my normalization process. So because I've binned in log space, and we don't live in log space, so then when we look at this if we were then to transform it into non-log space, there's going to be the bin widths vary. So I normalized each bin by their non-log space bin width. So that has an effect on parent slope. But this is because I looked at it the same way throughout the entire data set. While it's not like directly comparable to either of those other studies, it's comparable to itself. So I guess to make a long answer short, it's the methodology I used.
Not the fact that it's coastal versus open ocean.
I imagine that would have an effect as well, but I would hate to compare the two.
Okay. Yeah, it'd be interesting to see.
I have a comment there. With what you're explaining there is the standard way of normalizing, and then the slope should be comparable to what other people usually do. Yeah.
Other questions? No. In that case, let's thank Quinn again. Thank you very much.
Thank you.